Hi, Dolly. This is Merritt. I just thought I'd make a quick movie with the two slides that I wanted to bring to the event tomorrow. So there are two basically. One is showing a bit of a diagram and outlining basically our goal of either of having an agent that what we hope can do is either is make new neurons to replace the ones that are lost in Parkinson's, or to actually be able to protect neurons that from the onslaught of Parkinson's disease. And so there's a whole host of genes and transcription factors that our agent called PM803 can help upregulate that we've seen in some testing. So we want to be able to try to work this so that we can actually put it into stem cells that can then be placed within the brain of patients with Parkinson's disease, or even potentially be able to reprogram those cells. So the other thing is that we may be able to take this agent and these same sets of genes that are important for making neurons also are important in adult neurons to help protect them from Parkinson's disease. And so there's a whole host of ways in which they actually work, but that's the main idea. And so we'd like to be able to deliver that to patients before they've lost a lot of the dopamine neurons to save the ones that are present. So we may be able to actually generate a single factor that does that's able to serve the broad swath of patients from Parkinson's disease. So there's obviously some potential for therapeutic and commercial benefit. And so what we're looking to do then is we broke down what our value of this is. And so in terms of when we've gone out and talked to people about what companies, what they want to have, what they're looking for, one thing that they want is to be able to make dopamine neurons more quickly or easily. It's costly to make the neurons. The reagents are expensive. You have to have a well-qualified technician make it. So if you have an agent that can help accelerate it, that would be really useful. And so that's actually, we've been talking with folks at Cell Dynamics International, amongst other places, and we have a partnership with U of M Stem Cell Core being able to look at this, and we have experiments that are underway. But we need to complete some of these pilot experiments to help de-risk the investments of these commercial and these commercial partners that we have will be able to make that leap into supporting our work. So our route to translation, basically, how can we get it into patients? How can we make a difference? Is licensing our startup. It's pretty much the route for many of these things. For neuroprotection, here we're wanting to prevent the progression of PD with a single agent. And so rather than relying on a whole suite of different drugs, and maybe we can just use one. And so right now what we have is NIH support, so there's scientific rigor behind what we've been doing. And a partnership that's ongoing right now with Patrick Brennan and Jeffrey Kordauer over at Van Andel and Rush. So those are high-profile investigators that we've been able to collaborate with and get excited about our work. But in order to help kind of de-risk this investment and this early preclinical stage, we want to be able to get resources for support personnel and for reagents to help accelerate the progress that we have. So to help kind of heat up this kind of progress that we've been making with the NIH support that we currently have. So the route to translation there is, we have to get the IP around whatever results we actually end up getting from these experiments that we've been setting up, and then some licensing and or a startup for there. So so that's the heart of it. I think that will help. I just wanted to kind of make a quick video that might make it easy to see that.